So starting a unit on cellular respiration, and what you probably remember from Biology 1 is that respiration is where we break down carbs for energy, right? Very different than respiration just meaning breathing. We're really going to see in this unit why you have to have oxygen. But focusing on this idea that cellular respiration is going to give the cell energy. So before we do anything else, we really want to understand what is energy and how do living organisms kind of harvest or harness this energy. So if we take a second and we just talk about what energy is. Energy comes in lots of different forms. Main types of energy that we're going to see are radiant energy, which is from the sun, thermal energy, which is also from the sun, but can also come from chemical reactions. But we really are actually going to focus on potential energy and kinetic energy, and how it has to do with chemical energy. So this is kind of a good image to talk about kinetic energy versus potential energy. Kinetic energy would actually be for us like the movement of molecules. And here they're kind of showing that like you're having to put in work to get to the top of the hill. Potential energy is kind of like the idea of stored energy. It's there, it can be ready to be used, but we're not currently using any energy. So being at the top of a hill, you have this energy that you can go down. So kinetic energy kind of has to do with putting energy in or releasing energy. And we have some terms for these. Putting energy in is going to be endergonic. Releasing energy is going to be exergonic, exit. These are usually going to be building reactions in science. Exergonic is mostly going to be breaking down. So we talked about enzymes, we talked about this a little bit. The idea of synthesis reactions and digestion reactions, or anabolism and catabolism. So just kind of knowing, okay, what is kinetic energy? It's actual movement versus potential energy usually has to do with stored energy. So when we look at living organisms, all living organisms actually use ATP, which is adenosine, adenine, and ribose gives us adenosine triphosphate. All organisms use ATP as their energy storage molecule. Except this form of energy is actually going to be very, very short term. These bonds here are fairly easily broken. So ATP is not a long-lasting molecule. It's small, so it's fairly portable. It can actually easily be recycled. So if I take ADP and I add another phosphate, I get ATP. And this is storing energy. I'm building something, I'm storing energy. When I break this down, then I can go backwards and release energy. So ATP is what every living organism uses as an energy storage molecule. And the reason that it's a good energy storage molecule is these phosphate groups, these PO4 negatives, are pretty highly charged. And when I bond them together, there's a lot of potential energy here in this bond. When I break that bond, that energy can actually do work. So that's what we want to look at, is how do living organisms use energy? So we're kind of storing the energy temporarily in ATP. And then we're going to use this ATP to do work. And most of the work that we're going to be doing is movement of a protein, which we know is a conformational change. So organisms are going to need energy to move things, to build things. So either movement of something, or most times when we're building, we're going to use that energy. And so ATP, I know it's kind of hard to see in this diagram, but ATP is really used to kind of help move a molecule, that conformational change. So we're storing energy in ATP, every living cell, and then that ATP is used to cause some sort of change, either to build something, or a lot of times, for example, if you're familiar with the sliding filament theory of muscles, this is actually how a muscle moves, is we move a protein by putting an inorganic phosphate on that protein, and that causes a conformational change. So really all that ATP is a potential energy source that we can then use that energy to do work inside of the cell. So where does ATP come from? The main source of ATP is going to be through respiration, cellular respiration. And here's kind of a basic, straightforward explanation of it. You eat food, you digest food, the monomer gets broken down, and we get these products. So you're probably pretty familiar with this equation of carbs and oxygen getting CO2, H2O, and ATP. So what we really want to do to start this unit is really understand what is the actual chemical reaction. So we want to take a little bit of time and review what the chemical reaction is. It turns out that this is going to take place in a bunch of different stages, but we also want to know the overall idea. So the overall idea of respiration is that we take the energy in the sugar. Where is the energy in the sugar? The energy is in the bonds of the sugar. So we take the energy that's holding the C's, H's, and O's, C6H12O6, there's energy there, right? Where? Where is the energy? The energy is in the electrons, right? Energy is in the bonds. So we take the energy that's in these bonds, and we transfer that energy to a different bond. So energy is not being lost. It's not being created nor destroyed. That's that second law of thermodynamics. We're taking the energy in the bonds of sugar and we're transferring to ADP to make ATP. Adenosine diphosphate, adiphosphate on ATP. So we're going to see that oxygen is actually going to be the final electron acceptor. We'll talk more about what that means later as we go through. And then adding electrons is going to attract hydrogens, and that's how we end up with water. And then we'll talk about how as we break these down, the C's and the O's usually bond to make a carbon dioxide. So main idea of respiration. We take the energy in the bonds of the sugar. We slowly break the sugar down to store the energy in ATP. And along the way, we end up making these two waste products. So we want to do a quick chemistry review of when I'm transferring electrons. The transfer of electrons is called redox. Reduction and oxidation. Redox, reduction and oxidation, and they always go together. Reduction, remember, is gaining electrons. Oil rig, reduction is gaining, actually become more negative, become reduced. Oxidation is losing electrons. I become more positive. And so as I'm moving these electrons around, things are going to get reduced or oxidized. So we're going to need to be able to use these words, reduce and oxidize. You really don't need to worry about this top part. What we need to look at is kind of the picture. Okay, I've got a compound with electrons on it, and I transfer the electrons to this compound. So B is getting reduced, and it now is more negative. A is losing the electrons and is becoming more positive. Because what we're going to see is that we're going to take the electrons away from sugar. So if sugar is losing electrons, sugar is becoming oxidized. We're going to have electron carriers. They're going to carry the electrons, we'll see in just a second, to our electron transport chain. And the electron carriers are accepting the electrons and will become reduced. So being very familiar with the terms oxidize and reduce is really important. And knowing that those electrons have energy. So keeping in mind that electrons have energy. And so I take the energy from the sugar and I'm going to carry it to a place called the electron transport chain, and that's where we'll actually make most of the ATP. So the final part here is really going to be looking at all of the different parts. What we're going to see is that when we make ATP, there's actually two ways. Substrate level phosphorylation. All that phosphorylation means is add a phosphate. That means that I go from ADP, I add a phosphate, a little inorganic with that little I, and I get ATP. Substrate level means that it's happening by directly transferring. So we'll talk more about substrate level later. And then we've got oxidative phosphorylation. We'll talk about this more as well. But what we want to know is that there are going to be a couple main stages of respiration, and that we're going to make ATP two different ways: directly transferring phosphates, or this other way that we're going to call chemiosmosis. So keeping in mind, the big idea is this is all about transferring electrons, transferring the energy of the electrons. And so this is an electron transport chain. It's in the mitochondria. It's actually on the inner membrane. So the inner membrane of the mitochondria has these embedded proteins. Well, we're pretty familiar with that. And these proteins are actually going to pump hydrogen ions. We're going to create a gradient, and then we're going to use that gradient to make ATP. So the big idea here is that I use the energy of electrons to eventually make ATP. But we're going to use hydrogen ions, we're going to create a chemical gradient, and we're going to see that that results in the production of ATP. So lots going on here in this unit, but keeping in mind that it's all about taking sugar, breaking it down. Glycolysis is going to be our first stage, and glycolysis, we know glyco has to do with sugar, and lysis is actually the breaking down. So what we're going to see in respiration is that we very slowly break down sugar, and we end up with the energy being transferred to ATP. We will talk about fermentation, which is when we do respiration without oxygen. And so these are kind of the big ideas of cellular respiration. The transfer of electrons from sugar to a form of energy